I'm very excited to have Ulrike Bauer today on the Natural Reward podcast. She published recently a fascinating paper in Science magazine on the evolution of composite traits in pitcher plants. Ulrike, the reason I started this podcast partly was that I wanted to be able to talk to people in science without going to meetings. Sort of treat it like going to a conference, but being able to choose who's going to the conference. And I would kind of like to treat this sort of like I'm coming by your poster. Maybe you're explaining to me what your study showed, and at the end we can get to some more general themes. And I'm very interested in this topic of complex trait evolution, which is why I'm coming by your poster. and I'm very interested in the background of your study, and why you think it's important, especially, which we can get through what you actually showed, but I'd also like to get to some of the more general implications. But just to get to the background, what is a pitcher plant?
Good question to start with. A pitcher plant is one type of insect-eating plants. So there's a lot of insect-eating plants in the world, maybe surprisingly. And a pitcher plant is just one type of those. So it's a plant that uses a pitfall trap in the broadest sense, like an open vessel filled with fluid to capture insects and digest them and take the nutrients out.
And so how many species are there? And how many times has this evolved, do you think?
So it's
Just
evolved
ballpark.
multiple times.
Yeah.
There is probably close to 200 species, 300 species, somewhere in that region of pitcher plants. But there's not only a lot of species, there's also 3 or 4 completely separate groups of pitcher plants that have completely separately evolved over time. And by separate and unrelated, I mean as unrelated to each other as you and I are to a flatworm. So really absolutely opposite ends of the natural system.
And where do you find the pitcher plants?
All over the world, really. The ones I work with are distributed sort of around the Indian Ocean, broadly speaking, from Madagascar all the way around to Southeast Asia. But there's also pitcher plants.
I think most people in the U.S. have never seen one. I don't
No,
think I've ever
most
happened
people in
upon
the US will have seen one because there's also native pitcher plants in the US. But as I said, these are totally unrelated to the ones I work with, which are the Asian pitcher plants. But there is US American pitcher plants, there is South American pitcher plants, there is a totally isolated group of pitcher plants with just one species, which is only in the very western tip of Australia. So there's some bromeliads in South America and Middle America that also class sort of as pitcher plants. They look quite different, but they use a similar principle of having a water-filled tank in the middle of the plant, which also captures insects. So the idea in nature to capture insects with something that has a fluid at the bottom and slippery walls in some sense to be a good pitfall trap has evolved multiple times in multiple lineages.
And is there typically a bait, kind of like nectar or something to attract? What is it that attracts the insects?
Usually it is some sort of bait. Usually there is nectar involved. In the Asian pitcher plants that I work with, there's certainly quite a lot of nectar involved. And the nectar isn't just randomly secreted all over the plant, but it is secreted in higher quantities closer to the trap opening. So it's really a lure where insects find lower quality and quantity nectar dotted around the plants, but then if they go to the most dangerous places, there is higher amounts of more sugary nectar.
And in this one that you studied in the present article, it's a kind of complex spring trapping mechanism, whereas normally, do they just fall in?
Yes.
It's not as... Okay.
Yeah, normally, in most pitcher plants, it's really just like I said, there is some sort of water-filled cavity. It's got slippery surfaces either along the rim of that open cavity or on the inner wall, somewhere where the prey would slip into the trap. And it's usually, people have called pitcher plants passive traps because nothing is moving. Whether that's passive or not is debatable because the plant still does quite a lot of active mechanisms of attracting, digesting. So it's still alive and it's not just something passive that sits around. But there is normally no movement involved, apart from these two species that we've studied, where there is fast movement involved in trapping. So the trap is kind of covered by a roof-like structure that we call a lid, like in a pot. And that roof-like structure is normally not really involved in trapping in most pitcher plants. It may be attractive, but it's usually not involved in trapping. It's basically just a rain roof that prevents the trap and the contents to be diluted and to be flooded during tropical downpours. And in these two species, that's changed. And the lid has adapted a function in trapping. So it works like a springboard, like you say. It acts as a springboard, but it's not a springboard that's actuated by ants jumping on it, like you might imagine. But it's
so it's an interesting. It's sort of like the Venus flytrap, except it's passive. It's passive, but at the same time, it's allowing for a kind of somewhat active mechanism that's activated by the raindrop, right? So the plant isn't having to expend energy. It's kind of an ingenious mechanism because it's utilizing the raindrops energy to force the ant into the pitcher.
Exactly, it's an externally powered movement by using the kinetic energy of falling raindrops. And for multiple reasons, I really don't like this whole distinction into active and passive traps, because I think it's just distracting from the topic, right? It's really about moving traps and non-moving traps. Some things are just there and are sticky like a fly paper, nothing moves. And some traps employ movement. And whether that movement is actuated by the plant, by the environment, whether that's something that needs an actual physiological activation, like in the Venus flytrap, or it's something that is a release of stored energy, like in a spring where you first pull the tissues and fix them in place, and then something breaks and it springs out, like a lot of seed dispersal mechanisms in plants. It doesn't really capture this active-passive distinction. It's really about movement.
But if you're comparing it to if you're comparing it to the Venus flytrap, there's costs and benefits. I mean, the cost is you have to wait for it to rain and hope that a raindrop hits right at the right moment, whereas the benefit is you're not having to expend any energy or set the trap, supposedly. I mean, because like a Venus flytrap has to open every time it closes its trap, it has to at some point open it again, right, and then be ready to activate at the right moment.
Exactly, it's a trade-off between saving energy versus having more control, right? Okay. So the Venus flytrap is in much more control about when things open or close, when it's activated. The pitcher plant has no control over whether the trap is activated or not. As you say, it's just opportunistically waiting for a raindrop to fall at the right moment. But that's a very cheap way of going about it. So there's no costly physiology, growth processes, opening processes, all of these things involved. It's just once the trap is there, and it's ready to trap. And it's not the only trapping mechanism of that plant, right? The slipperiness is still there. So it's just an additional income, if you want to.
Do you have any photos of pitcher plants that you would like to share?
I do. Let me see. So I can maybe show you some generally some photos of different types of pitcher plants. Let me see if I can share just this window. Yeah, you should be able to see this. So this is the Australian pitcher plant that I've briefly mentioned, Cephalotus, which is really just an odd one because it's one single species in one single genus. in a completely different order to all the other pitcher plants, just on the very southwestern tip of Australia. So a very weird one that's evolved just completely separately from everything else. And then if you compare with the South American Heliamphora pitcher plants, and this is actually a Nepenthes, so an Asian pitcher plant, you see that the structures are really similar. The pitchers are very similar. So even though they're completely unrelated, they've come up with quite a similar solution to trapping. And I don't know, can you see my mouse at all?
Yeah.
Yeah, so you can see the rim here of the pitcher, which is usually very slippery for insects, especially when it gets wet. And then you also get, so you can see that within Asian pitcher plants, there's actually quite a lot of variation in shape and size and the way a pitcher is built. So this is still an Asian pitcher plant, but the pitcher looks very different. You don't really have this rim at all. You have a different shape. You have this lid being just a thin strip which stands up, so it's not really a roof anymore, while here you have a big roof-like lid and a massive rim. So
why does the lid exist in the former one you just showed? That
this
looks
one.
Almost like, yeah, why does that one have a lid, do you think?
Good question. Why it has a lid. The lid is secreting nectar in this one, so you often see insects coming to the lid. What the lid does, you can only speculate here. It definitely doesn't serve a lid function anymore. This is actually a species that has presumably a really weird, unusual trapping mechanism again, so it probably works as a flypaper trap. So the fluid that is in this species, and you can see a lot of prey accumulated inside the bottom here, the fluid is very, very sticky, like a very thick viscoelastic slime, and it doesn't seem to mix well with the water. And because there is no lid, water will just fill the trap when it rains. And eventually it will tip over because it gets very head heavy when it fills up, as you can imagine. And the water runs out, the sticky fluid stays in the bottom because it doesn't mix very well and it's very sticky. But a little bit of sticky fluid coats the walls as it runs out. And so this works as a flypaper trap. So when it's not filled with water, small flies, small insects get literally stuck on the walls here. And I think there are some stuck to it, which are barely visible. I think there's one here on the inside that you see the shadow of. When it rains, they get washed into the fluid at the bottom. The trap tips over and kind of resets itself. So it's an example of another odd one out that's come up with a different trapping mechanism to the rest. And there's quite a few within that Southeast Asian group of pitcher plants that have evolved away from the normal pitfall trapping and trap different things, different ways. So it's a very very diverse group in terms of shapes and trapping mechanisms. So yeah, you can see here the diversity of shapes and models that evolution came up with. And then this is the North American Sarracenia pitcher plant that maybe people in North America are familiar with, especially sort of like around the northern edge and eastern parts of the US. You find quite a lot of these. This is actually here in Hampshire in the UK where they are invasive. But I have seen them in Massachusetts and also in Canada, just growing in the box.
Describe this one. It looks a little different. It looks like a little Red Riding Hood or something.
Yeah, it is a little different, but it's still a kind of very similar. Hang on, can I actually zoom in here? Yes, I can. So you can see the pictures a bit better here if I zoom into this a little bit. So you can still see it's still sort of like an opening. It doesn't really have a lid in the way it has a hood. It has these downward pointing hairs here, which are presumably slippery. So it's a similar principle that you have a leaf that's transformed into a fluid vessel with an opening, and insects fall in and get digested at the bottom. So even though it's a different group to the Asian one, so the pitchers are slightly different in the way they grow, right? They grow as a rosette. They grow straight out of the ground, so you don't really have a leaf part to it. You only have a pitcher. And the only bit that acts as a leaf for photosynthesis is basically this keel on the side of the pitcher.
Huh. And so do you consider the pitcher a key innovation? Is it something that's triggered diversification, the ability to invade novel habitats? Clearly, it's something that gives you the ability to tap into a new resource zone, right? By
Definitely.
The ability to...
Definitely. The ability to trap and digest insects has evolved multiple times in plants, and not just through pitchers, but also through other mechanisms like sticky leaves and like suction traps and all sorts of different eel traps, all sorts of different trap designs have come up.
Yeah.
So there's clearly an advantage to capturing insects if it's in the right settings, in the right habitat, right? So carnivorous plants typically grow in places where there's lots of light, lots of water, but a lack of mineral nutrients.
Okay. And then these pitcher plants, there have been, as you mentioned, multiple independent origins. Have each of those origins been involved with a diversification event, with a expansion of the number of species, or are there examples where maybe there's only a couple of species, or did it always lead to like 50 species?
No. There's examples where there's just one, right? The Australian pitcher plant, for example.
Oh, okay.
So that's just a single species, which is quite odd, right? There's in some instances you have a huge diversification, like in the Asian pitcher plant, which probably are somewhere between 150 and 200 species on their own. And in other cases, there's fewer species.
And the one you studied in this paper, how many species are there?
Around 150 to 200. It depends a bit where you stand as a taxonomist, what you consider a different species or the same species. Like
what is that clade called?
Nepenthes.
Nepenthes, okay.
Nepenthes, Asian pitcher plants. Or I think people say monkey cups in as a sort of trivial English name.
And so this trait that you studied, the spring trapping mechanism, you describe as a composite trait, meaning that it has multiple components. And I was just wondering, do you see the pitcher itself as a composite trait, even without the spring trapping mechanism?
Certainly, yes. There you need, again, multiple independent adaptations to come together to turn a flat leaf into a pitcher. So you need to change in shape, you need to change in physiology. You don't really need to invent the ability to take up nutrients, because leaves can usually take up mineral nutrients when they're dissolved in water. So that's the simple reason why you can spray fertilize your plants. And it works. If you just spray your leaves with diluted fertilizer solutions, they can take up through the stomata, through the breathing openings in the leaf. They can take up nutrients. But the ability to secrete something or bring up a substance that traps insects, that retains insects, to secrete enzymes, to digest insects. The secreting fluid into a cavity or secreting sticky fluid on tentacles, as you have in the sundews. So making a carnivorous leaf is a very complex modification of a photosynthetic leaf.
So that's a more complicated scenario than the one you're studying. So in this case of the spring trapping mechanism, it's a composite trait, but it's not so complicated that it would be impossible to, or it makes it more amenable to a study like yours, where you're looking at each component part and you can split it into 3 traits and look at how each varies and get an idea about what happened in the history of evolution. So how about letting us know how you designed this study, and I'd really like to know kind of how you got the idea to look at this as a composite trait, and then kind of just what your study showed.
Yeah, so the ideas really evolved over time, almost like the traits. And I think that's quite a common thing in our research, that you don't necessarily set out and you have all the ideas laid out in your head, but you go out, you discover something, and then you kind of gradually or come from one question to the next as you study your system, right? So we initially just studied this trapping mechanism in one of the two species, in Nepenthes gracilis from Borneo, because we happened to discover the trapping mechanism about 12 years back in Borneo, and then really wanted to understand how it works. So I'm not actually an evolutionary biologist by training at all. I'm somewhere at the intersection between ecology and biomechanics, and I'm usually trying to understand how things work from a mechanics point of view. So I'm really interested in plant surfaces, in plant structures, and plant materials, and how they work together and interact with the environment to assume a certain function, in this case being an insect trap. So I worked for about 5, 6, 7 years on how this springboard trapping works in Nepenthes gracilis, and found for that species that there are three components that need to come together for this to work. So the first one is really trivial. If you want to work as a springboard, you need to have your spring oriented so that the prey lands in the cavity where you want it. So in that case, if the cavity is below, you need ideally have your lid horizontal, right? Because if you have it at a 45-degree angle, the insect will still get kicked off, but it will miss the trap. So that's really trivial. Secondly, you need spring properties, because if it's really wobbly, and I can show you a video to demonstrate that, actually. Let me. So if I play this again, you can see that this lid really is very springy. It really vibrates really stiffly. While if I... Does this work now?
Yes.
Yes, so this is a lid that doesn't work as a springboard, and you can see how it's really wobbly and really not working as a spring. It's just sort of wobbling about when it's being hit. So you need the spring property to transmit enough of the raindrop energy to the prey to actually kick it off. If you just wobble about, there's not much energy being transferred. And then thirdly, you need to make the underside of the lid somewhat slippery, because insects are really damn good to hold on to things. So if you see a fly walking upside down on a glass plate, you see immediately how good insect feet are at sticking. So if you had a perfectly smooth surface, you could hit it with a raindrop, the insects would not fall off. And we've done that experiment. We've manipulated the underside of the lid to make it really smooth, and the insects did not fall off anymore. So what you need is a specialized wax crystal layer that makes the surface just slippery enough so that insects can't hold on when they're being hit, but not so slippery that they cannot access in the first place. So you have wax crystal surfaces in plants that are incredibly slippery, like on the inner wall of the trap, where insects can't get a footing on at all. And if that was under the lid as well, they would never go there, so they would never get trapped. Yeah. So you need this specialized coating for insects to be able to just go there, but then still be kicked off. So we spent a couple of... Sorry, yeah, go ahead.
What was the second species that you found this same mechanism in?
So the second species is Nepenthes pavillii from the Seychelles. And that was, again, a pure coincidence that we came across that. So I was on a private trip to Madagascar in 2016, had a stopover in the Seychelles, and decided to stop for a couple of days rather than just a few hours, because I knew there is this endemic Seychelles pitcher plant, which I wanted to see. So I stopped there with a couple of friends, went up some mountains, found the pitcher plants, and got really excited seeing that they also have wax crystals under the lid, and they also have a horizontal lid. And when I prodded the lid, it had quite springy and stiff properties. So I was like, this in theory really has all the traits it needs to be a springboard trap. I wonder if this is also a springboard trap. And so I came back half a year later to do some field work. And indeed, it works also as a springboard trap. And this is really where this paper started, right? And where initially we didn't really think about how this can evolve. We just thought about, wow, there's two species, one in Borneo, one in the Seychelles. They're not direct sister species. They're both in the same genus, but they're quite a bit apart in the evolution. And we don't know of any others that have a springboard trap. So there's these two guys come up with the same crazy mechanism. While before I always thought like this is a one-off, right? A mechanism that needs three things to come together in such a constellation, with wax crystals under the lid really not being a common trait. I thought, well, this is a crazy one-off that like the flypaper one I've shown you earlier, the one that has the tip-over mechanism with the flypaper trapping. That's just one species that does something different and something crazy. And suddenly we found a second one. So that's where my head started turning, and I was like,
and
okay.
You had just discovered this first one or described it in around 2017, right? Or
the first one was earlier. The first one was around, I think the first paper was in 2012.
Okay. But this is something that you had worked on, and did you discover the first one?
Yes, we discovered the first one during fieldwork in Borneo. And again, just by like being out in the field, observing a few things, putting two and two together to make a story, and then doing experiments to confirm what's happening.
You were the perfect person to notice this because you had noticed the first one and described it, and so you're prepared to see it again.
Yeah, in that sense, yes, but I think you also, it's just being out in the natural environment with an open mind and open eyes, right? And looking at things properly. That's how you discover things in general.
True. But you knew now that you knew what the spring tracking mechanism was, you would be able to see it quite easily compared to somebody that had never thought about it before.
Yeah. Yeah, for sure.
But
then you don't see it in action, right? You don't see it. That's the problem. You don't observe it in action in nature because it's still a rare event. You have to look in the right moment, and it happens incredibly quickly. So with the naked eye, you don't really see the insect falling. The spring is fast, right? So you just the most you would maybe observe is that there was an insect, and a moment later it isn't anymore.
Okay, so then how did you put a team of people together to work on the evolution of this? Or how did the group of people come together?
Bit by bit again. So with every question that came up, I involved another person into the study, really. So initially I just went on fieldwork with a colleague of mine, Beth, who is a co-author on the paper, and we decided just to investigate this trapping mechanism in Nepenthes pavillii. So even figure out, does it really do it, and how does it do it, and do vibrometry measurements or analogous to what we had done with Gracilis before, just to sort of compare, do they really do the same thing? Have they really evolved the same mechanism, or have I just observed something and my mind ran ahead of me, right? And so that's how Beth came on board. She just happened to share an office with me and have a vibrometer. And so I said, well, do you want to come and do fieldwork in the Seychelles with me? And a few people will say no. It's a great place to do fieldwork. Not the cheapest place to do fieldwork, but a really nice place to do fieldwork. And then I had been in touch with Reinhard Jätter's group in Vancouver before, because they helped me to look at the specialized wax crystals under the Gracilis lid. So I asked them if they would be happy to also analyze some waxes from pavillii for me. And so I just sent them wax samples. So 3 of the co-authors on the paper are from that wax group and have all worked on the wax analyses. And the the idea to look into the evolution of all of this evolved really over time, because we were sort of like thinking, okay, the paper could go in the direction, is it convergent evolution of the two trapping mechanisms in the two species? So did it really evolve twice independently, or is this something that is inherited from a common ancestor? And I'm not an evolutionary biologist. I didn't have the toolkit to distinguish between these two ideas. So I searched around to find someone who does evolutionary biology, and I knew Guillaume from meeting him at conferences, and so I approached him, and I think I even came and gave a seminar at his uni. I think that's how I got Guillaume involved. I gave a seminar at Durham University in 2018 or 19, and talked about this work with him. And he said, like, oh, well, I can help you to figure out if it's convergence or not. And from there, we really then continued working together. And the idea the realization that we're talking about something bigger here than just convergence or not, but that we have an opportunity to look at how such a composite trait can evolve, because we have the two species that evolved it independently. That was something that came up like way later in the discussion, how we spin this paper. And we were thinking, okay, like how can this happen? Come up with hypotheses. And then it's just one step further to think, how can we test these, right?
Yeah, so I can't wait to get into this evolutionary part because that's my main interest. But I just want to go back for a second. Your original trip to the Seychelles was a personal trip, right?
Yeah.
And you just did this in your spare time. Did you have any expectation that you would find the spring trapping plant? I mean, obviously no, right? Because you thought it just evolved once. But in the back of your mind, are you like looking for this in places? Are you thinking, I'm in this new habitat, there was some reason you might find it there, or was this just random?
I think it's somewhere in between, really. So I don't go out expecting to find anything, but I've been working on pitcher plants, and especially Asian ones, for 2 decades. And I'm always curious, and I always look at them closely when I see them. And if I have an opportunity to see another species in the wild in its natural habitat without like going totally out of my way, I will take it. So I've over the years tracked down about 80 something species in the wild, and I'm constantly looking to add to that. So if I have a chance, if I stop over in an island anyways that has a pitcher plant species fairly accessible that I haven't seen before, then it's a no-brainer to me to stay for a couple of nights and try and see it, right?
That's really great to have an excuse to go hiking, isn't it?
Oh, always.
Yeah. Yeah, that's really cool. Okay, so I was just wondering also if you had any photos of that trip when you found this plant. Did you take any photos of it when you were looking at it out there, or?
Yeah, I did. And we took lots of photos generally. So I actually met up with a couple of carnivorous plant nerds there on that occasion, who all wanted to go to Madagascar. And so we just all decided to come a few days earlier and stop in the Seychelles and just see some botany in the Seychelles, right? Not just carnivorous plants, but generally like some of the oddities that grow there. So yeah, it's a group of friends that I've traveled with for nearly 2 decades to various carnivorous plant locations around the globe, and had some great adventures with.
Would you want to share any of those or no?
Where to start and where to stop, really. It's a bunch of people. We originally.
Oh, no. Sorry,
I mean,
I just meant like share a photo from that trip.
let me see if I have any. Because I struggled a bit to find too many photos. So I found a few photos from the Seychelles, but I think they're actually from the field trip that I did the year after. So let me see. but it gives you an idea how the plants grow in their natural place. So this is sort of like Nepenthes pavillei growing on granite outcrops on Mahe Island in the Seychelles, and they form these like creeping carpets of pitches, really, really beautiful. And this is on the neighboring island on Silhouette, right near the highest peak on that island where pavillei grows, and you kind of like look down into the ocean behind.
So did you first see it in a environment like this where you're up on a hilltop, or?
Yeah, they all grow up on hilltops, so they all
Okay.
grow slightly up on sort of like exposed outcrops, and yeah, you can see here close to the highest peak in the Seychelles, just forming a carpet again, and that's sort of like in the field side, me doing a selfie with lots of pitcher plants behind, actually in my main field side where I did most of the experiments for the paper. So
That's really cool.
it's a really cool place. It's a lovely place to work, and this site is actually not a long hike at all. It's in an old tea plantation, and it's like an abandoned tea plantation, and it's literally a 5-minute walks from the street. So it's very convenient, and yet you don't feel like you're right next to a major road, right? Mind all the major roads in the Seychelles are fairly small and insignificant, really. So yeah, it's a lovely place to do field work.
Yeah, it looks really nice. Okay, so you're not an evolutionary biologist, but you happen upon this independently evolved trait that's complicated. And so, what did you immediately think might be the case?
Well, we initially started scratching our heads, really, because we were like, how can something like this evolve when you have a new trait, springboard trapping, which only works if you have all three components in the right constellation coming together. So each of these components on their own don't have a function for trapping, right? A horizontal lid is pretty meaningless unless you have the other two, the springboard properties and the somewhat slippery coating with it. It doesn't
It's
work
sort of
as a
like
trap.
it's like the original irreducible complexity problem. Irreducible complexity, meaning like you got to have these different parts for the whole thing to have an emergent function.
Exactly.
And
exactly. So these are like the three essentials that you need, but each of the components on its own doesn't necessarily have a function, right? So there is the question, how can something like that come about if traditionally we think of something more complex emerging because the components get selected gradually and gradually evolve from something more simple into something more complex. But if this relatively simple but composite mechanism is really a novelty that only comes about when you have these three components and beforehand they don't really serve a function, an easily conceivable function, then how do you get there without them being selected in a certain way? And so initially the question was just, well, do we have a selection for something else that drives these components towards that springboard trapping for some reason, something that we don't understand, don't see, don't know about? Or are they like, is there another way how this can come about? Like, I mean, the obvious other way is just a chance encounter. A lot of things can happen just by the right constellation just coming up by chance, like in a giant evolutionary lottery. And in that case, you increase the chances of that happening by increasing the number of tickets in the lottery. Right. So we had our two hypotheses to test. Do we see a gradual modification of these three traits together in the right direction? Or do we not select these traits at all? Do we allow them to just fluctuate wildly because there is no selection, so there's no pressure for them to go in any direction, so they can do whatever they want. They can be really variable, And by making them really variable, we just increase the number of possible combinations and increase the chances of our beneficial combination coming together. And so we went out to test those two hypotheses. And
so did you, before you talked to Guillaume, did you have those two separate hypotheses, or is this something that you developed with him talking about it?
This is something we actually developed really as a team, so beyond just Guillaume and me, but also talking to Reinhard, or mostly to Reinhard. It was really mostly Reinhard, Guillaume, and me having a lot of discussions, or I think Luke as well at times. So we used to set up Zoom meetings between us and from time to time discuss ideas and discuss where we see this paper going. And then at some point when we kind of like went beyond the immediate, we would just want to see whether these two species really have the same mechanism. When we really started thinking about evolutionary mechanisms, then at some point it ended up being more a work between Guillaume and me. But we always talked about things. We always bounced back ideas back and forth. We met up multiple times, both in person and online. And I think initially things were really significantly triggered by talking to Reinhard as well, who is a biochemist. So he's really not an evolutionary biologist, but he's amazing in the way he thinks about things. And he asks a lot of questions, really sort of interest-driven, curiosity-driven, but good, insightful questions. So I think these questions that came from Reinhard, whenever we talked about the story, really helped Guillaume and me initially to come up with these ideas, because if someone asks the right questions, it's amazing how it prompts your brain to think, right?
Interesting. I'd just like to mention an example of a complex trait that you often read about in evolution textbooks. I've noticed that most evolution textbooks have about 4 pages devoted to complex trait evolution, and almost all of the pages are devoted to the eye, the evolution of the camera-like eye, right? And this is I don't think the best example of complex traits in general, because from the simplest eye spot to the most complex eye, you can explain the subparts of an eye as having been either initially originated or co-opted for the purpose of vision, of detecting spatial information. And there's not like room in there for these kind of alternative ways that you can get multi-component emergence happening, where the causes for the origin of some multi-component trait is different than why it is then selected later. And so let's just get to your study, and then I'll mention an example that I think is similar to yours in some ways, and what the common theme I think might be between the two examples. Because in this other example, it's not that the traits originate randomly, but there's still what I would call multi-component emergence, or what you might call the composite trait, where there's an emergent function that comes from these parts. But in some cases, it's not totally random that they come together. But what's the evidence that the different traits in your case came together by spontaneous coincidence rather than selection?
So if they had been selected together, we can make 2 predictions what we would see if they had evolved together and gradually, right? We would expect that the evolution of these 3 traits usually occurs together in the phylogeny. So we see them either all emerging at once or not at all. We wouldn't see them just one here, one there, popping up in the phylogeny. So we would see some sort of correlated evolution when we test for correlation of the evolution and emergence of these traits across multiple lineages. And we don't find that. So you can test for that, and there is no correlation in the way they evolved or not evolved in different species. The other thing we would expect for our spontaneous coincidence is, as I said, the idea behind that mechanism is that you allow the components to be highly variable so that you have a larger pot of lottery tickets to draw from. So we would expect this increased variability in the species that don't have springboard trapping. And the devil is in the detail here. So if there was selection in each of these component traits for a state that we would need for the springboard trapping, then we would expect that variability to be reduced as soon as a species has the right adaptation, right? So as soon as we see a lid approaching horizontal, we would expect that if selection is there for horizontal lids for some other reason than springboard trapping, we would expect horizontal lids to be less variable than other lid angles. So species that tend to have horizontal lids would show less variation of lid angle than species that have 45-degree lids. And we don't see that. We only see the reduced variation in the species that actually use the lid as a springboard. So that indicates that stabilizing selection is only there when we have the new function. It's not there in the individual components before we get the new function coming together.
Yeah, that's pretty nifty. That's a kind of clever way of providing support for this idea, or testing it, I guess. I mean, because you could have found the opposite, in which case you might have concluded that there was selection, and that's it. So let's explore the alternative scenario that you might have found, which is that the horizontal lid is not variable even in the species without the whole spring trap. That would suggest that there is some selection for being horizontal that's different than the spring trap. And one thing I would point out is that if you imagine a scenario where each of these independent traits was stabilized by selection for some other reason that wasn't the springboard trapping, you could still get the composite trait basically originating from spontaneous coincidence. In a sense, it's not totally random because selection would be stabilizing, but not for the ultimate effect, right? The incidental effect is still random compared to why they were selected.
Yeah, so you could still get a new function by selecting traits for something else. So something that often is called pre-adaptation, right? You have something that is selected for a different function, and because it already assumed a certain state of that trait, it has, like people say, it's pre-adapted for a new function, right? So if we already have a horizontal lid because a horizontal lid is maybe a better rain roof, well, then it's already horizontal, so you only need to bring in the two other traits in the right constellation, which also leads to the second thought that you don't necessarily need all three traits to follow the same scenario, right? There's no reason why it could only either all be variable and coinciding, or one could be selected directionally, the other one could vary wildly, right? So we're not saying like necessarily it has to go that way. And actually for the spring properties, we have a mixed evidence that shows that it might be the variability, or it might be a mixed scenario where lids are actually selected for being springy, because we find some evidence that in some species with springy lids, there's also less variations. The lids tend to be all springy. This could be because there's an advantage to being springy, or it could be because there's a constraint coming with being springy, right? Being springy could be a side effect of just having stiffer material, being more durable, being more long-lived. So a trap could be selected for longevity, making the material more stiff and more durable, and that could help actually making it more springy.
Yeah. Okay. So it's a little variable with the lid, with the springiness of it, but the lid angle looks like it's random, right? Or it's more variable in the non-spring trap species. suggesting that it's not
exactly.
under
For the
selection.
angle, we only find strong evidence for this coincidence mechanism.
And
Basically,
then what about the wax?
I think the key thing is not necessarily being able to say, oh, this one is definitely totally going to be this mechanism or that mechanism. I think the exciting thing about the paper is that we show an alternative way how you come up with a novelty. So evolutionary biologists seem to think a lot about selection-driven evolution and maybe not so much often about the random components in there, while there are obviously papers that clearly show that randomness is important, right? Random effects can bring novelty just as much as anything else, and they're often neglected when people think about evolution. People don't think so much about
let's get to that in a second. I just want to ask about the waxiness. How does that accord to what you just said with the other two traits?
For the waxiness, we are actually not able to say much because we simply were not able to look at variability of the wax layer. We didn't have enough samples to stick into a scanning electron microscope to really quantify variation of wax crystal cover and morphology across a large amount of samples. And it's partly because you have to rely on botanical collections to get samples from all the different species. They don't tend to like if you chop off lots and lots and lots of traps and take lots of samples. And the other is the just time-consuming and costly process of imaging them in a scanning electron microscopy. So
but that's only part of your argument. The other part of it is showing that across all these species, both of these traits vary a lot.
So wax crystals generally across species are highly variable. We also have some evidence that wax crystals within the same plant are easily adapted because we see different types of wax crystals within the same pitcher on different surfaces. So the inner wall has different types of wax crystals to the lid in the species that have wax crystals on the lid. A lot of species don't have wax crystals under the lid. So physiologically, developmentally, wax crystals are something that is highly plastic and is very easy to change and adapt for plants. And the biosynthesis of waxes kind of sets them up for being really easily changeable because it's a modular process. It's basically taking a starting point and then adding building blocks. And you keep adding building blocks and you make longer or shorter chains. And at any point in these building blocks, you can just divert the biosynthesis and make a different compound, cross-link things, or turn them into alcohol. So you can modify them chemically really, really easily. And the biochemistry of the wax will determine what the crystals look like. So by just tweaking one little step in the pathway, you can turn one surface into an entirely different surface very, very easily.
Interesting. Okay, but I want to return real quick just to the main line of evidence. Let me get this straight. So you're expecting that if it's kind of a gradual natural selection, almost like hill climbing on an adaptive landscape or something, you would have the three traits almost as if they're different loci contributing to a quantitative trait, where they would all be kind of gradually being moved up to the same peak towards this purpose of the pitcher plant trapping. And in that scenario, it would for selection to work that way, it would be necessary that every trait would gain a little bit of an advantage by being modified gradually towards this complex trait. Kind of on the surface of it, it would be difficult for that to happen because each one of these traits has to be pretty fine-tuned to get the emergent function of the components working together. So you wouldn't really expect that an intermediate state of each one of the traits might have a emergent interaction with other traits that actually works very well.
Exactly. Exactly.
But if it were that way, and you have to entertain that for the purpose of testing the model, you might expect maybe an intermediate species that, is kind of a transitional species that lives somewhere in the world, and it's on its way to the pinnacle of the peak, but it's not there yet. And so you might expect some, you often see this in phylogenies where there's something that's new, a novelty, and then there's some forms connecting it to the ancestor that are intermediate between it and the ancestor, suggesting a sort of gradual evolution to that new state. But here you don't see that. You just see there's no transitional links like that. It's just the traits are just varying all over the phylogeny, seemingly in random combinations everywhere. And then where you get the right combination is where now all of a sudden, at least with the lid, it's being stabilized by selection now to be at the right angle, at the horizontal angle, whereas in other species, it's not being stabilized by selection, as suggested by more variation within the species that are like that.
Exactly. And then it's quite, from that, it's quite intriguing to think a step further and think that there are key inventions in evolution where, when we look into the fossil record, we actually don't find any transitional stages.
Sorry, I got to deal with my dog for a second. Give me 1 minute. Sorry.
Yeah.
Buddy, come here. Here he is. He got out.
Oh.
So there's pretty good evidence that this did not evolve kind of gradually, with all the traits going in the same direction, that the traits kind of, you've got this emergent property that came together because, like you said, there's a lot of lottery tickets with each of the traits varying independently, and then it just happens to come together in these two species. And you showed in this paper that it was independent evolution, right? That was part of what y'all did, was the phylogenetics, showing that it wasn't just one origin.
No, exactly. It's like very unlikely that the two species have inherited this mechanism, and all the other ones in between have lost it, right?
Yeah.
So all the evidence there points towards two independent origins.
Yeah. And so have you ever taken an evolution course as an undergrad, or kind of read, you know some about evolution. I mean, you know the history of like adaptive landscapes and this idea of
sure,
peak climbing that's so dominant. Okay, so how do you see your discovery here as relating to common, I don't know, what textbook dogma in evolutionary biology, or just what you've absorbed from your knowledge of evolution?
So I think the main surprising thing here is that we don't see gradual selection leading to something new. Like you brought this example of the mammalian eye, right? Which is such a textbook example of gradually modifying a structure to become more and more complex and more specialized. And there's a lot of examples where complex traits, what we talk of as complex traits, have originated by like gradually modifying something in a certain direction. But there is usually an underlying purpose already. Like you said, the early stages of the eye were already structures for light capture. There were already structures with a purpose of visually capturing the world around an organism. And so it's more a process of refining what's already there. And that sort of evolution that is taught over and over in textbooks. And it's of course highly valid. Like, traits are refined that way, like evolution works that way. Selection is a major driving force of evolution, but it really struggles sometimes just based on nothing else but nothing else but that concept. It really is hard to explain how entirely new things can emerge, how you can come up with something that has a new function emerging from some major new invention. So we find these novelties popping up again and again and again in evolutionary history. And like you said, normally it's things where we don't find any transition stages, right? For the eye, we find lots and lots of transition stages. For long necks, like giraffe necks, you find lots and lots and lots of transition stages where something got gradually elongated over time. For pitcher traps, we don't find any transitional stages when we look into the fossil history. You find plants that have a pitcher trap or plants that don't have a pitcher trap, but there's no extant transitional intermediate stage. There's no fossil intermediate. So it leaves evolutionary biologists with a great puzzle to solve. How did this come about if you don't find any fossil record of it gradually evolving? And I think this concept of thinking of things that they don't necessarily have to evolve gradually over time. Things happen in an instant sometimes. And this can be just a single mutation somewhere. So with pitcher leaves, it could be that it's a single mutation that fuses a leaf into a tube rather than having a flat leaf. And we know from other species that are not carnivorous that occasionally this can happen. There can be really a single mutation in a leaf that turns a flat leaf into a tubular leaf. But there's also the option that you have a more complex trait, like with the springboard trapping, where you need several components. And then the role of allowing traits to vary and the role of variability of traits really is gets a new meaning, right? It becomes more, how to explain this? It becomes a very important factor to have traits, to allow traits to be variable when you think about random effects. And I think random effects are generally not considered enough when we think about explaining the natural world, because we like to explain things by clearly defined mechanisms. We don't like randomness as humans, right? Randomness is something scary because it's chaotic.
Yeah, so I'd say a couple of things about what you just said, because I think there's a tendency to focus on saltational evolution, jumps, among people that have provided an alternative to Darwinism as an explanation for macroevolution, for the origin of complex traits and the major trends of evolution. People often talk about major leaps, that meaning sort of like what you just said, you can have these big mutations, that this would play a big role. And then the role of randomness. So my perspective first is that there aren't enough studies of complex trait evolution. So every example like yours is really valuable because evolutionists have focused on simple traits that are amenable to study in the laboratory, usually with a simple genetic basis or quantitative traits in populations. And there aren't many studies that have really been able to examine the evolution of complex traits. And those that have looked at this are often interpreting the fossil record or interpreting the data in a way that meshes with a certain way of looking at it. And there are only a few studies where people have actually been able to look at the sequence of events leading to the evolution of a complex trait and like actually known what happened. And I think your example is very detailed, and it shows just a nice instance where it's not according to the standard narrative. So the way I look at the origin of complex traits in general, like that I look at it as there is a big role for gradual evolution typically. I think in your case, it's a little bit of an odd case, but I think the connection is not that gradual evolution is not important, but just showing that how emergence happens and how the component parts typically have to be at a certain level to interact effectively to produce the emergent function. So you found that. And what we were talking about earlier, in your case, these individual traits weren't necessarily under selection for other purposes. They're kind of varying randomly, possibly. It's hard to tell exactly with 2 out of the 3 traits, but one of them looks like it's randomly varying. My point would be that in possibly a lot of other instances, the traits may be selected and may evolve gradually. And in some cases, you can have a sequence of events that... So let me just give the example that I studied, which is the evolution of kin recognition. And it looks like that this evolves through a sequence of steps where one step leads to the next and leads to the next. And at each stage of evolution, there's a particular selective pressure operating. And it's just in response to the immediate environment of the organism. And those steps gradually build a complex trait that at some point has an emergent function. And it actually, once it's there, it appears designed for a purpose that it didn't, which is different than the reason that it originated, and has this incidental effect that is different than the process leading to it. So it is a selective process. It looks like it's happened many times independently. And another example of that same sort of stepwise process would be the evolution of self-incompatibility in plants and animals. And in that case, which is a much more familiar, I'm intentionally not getting into the details of my model because it's still something that I'm trying to get accepted and people to start testing it and everything. But it is pretty well accepted that the evolution of self-incompatibility evolved through a similar sequence of steps where you initially had the evolution of some kind of self-mating or kin mating, as often happens in plants with hermaphroditic flowers, or it also happens in hermaphroditic marine invertebrates where they can mate with themselves. And then you have similar things with kin mating in higher animals. And well, what happens with self or kin mating is inbreeding depression, right? And so then that can have immediate deleterious effects on the offspring. That can then select for behaviors that avoid mating with self or kin. Now, once those behaviors are present, it can favor the evolution of the polymorphism of the cue used for self-incompatibility. So then you get this hypervariable genetic cue that allows you to effectively distinguish kin from non-kin. And then once this whole system is present, it promotes more outbreeding and minimizes inbreeding. And this can have long-term macroevolutionary effects on the rates of extinction. And that's been shown with phylogenetic comparisons where self-incompatibility has been lost and species have transitioned to self-compatibility. They then have higher rates of extinction and lower net diversification, potentially because the selfing species might not evolve as quickly as the outbreeding species, which is similar to an argument for the long-term maintenance of sexual reproduction, the increase of evolvability. Essentially, outbreeding is analogous to having sexual reproduction, whereas inbreeding or selfing is more like being clonal in a sense, right? Because you're potentially clonally propagating. So the difference, what's similar is, for example, in self-compatibility and self-incompatibility, is that you can have an emergent you can, well, let's say in that case, an incidental effect on rates of extinction. Okay, so you can separate cause and effect. In my example of kin recognition, there's also a potential incidental effect that's different than the cause for origin. In terms of kind of being like an emergent function, I don't know if I would like necessarily look at it in that way, because each step just leads to like a new adaptive situation that's gradually building a trait that becomes more complex over time. And it's not like there's just a threshold at some point where it's like your pitcher plant example, where all of a sudden there's like it's able to do something totally new. Each step is something new. But I think probably the similarity is that there's a potential incidental effect that's different than why it originated. So this would get to your example, and I would ask you here, I mean, we were talking about pitcher plants in general. The pitcher could be a composite trait that is a key innovation that in many instances led to diversification events. And so it's possible that the parts of the pitcher plants that came together, what you were talking about earlier, the different metabolic abilities and everything, okay, they initially evolve, and some of them must have been built through natural selection, because you're talking about some complicated chemicals, and even like you said, the leaf ability to absorb nutrients and everything, that ends up being important for the pitcher plant mechanism, being able to digest prey and everything. And so these different components were evolved by natural selection for these different purposes, and then they become part of this complex pitcher plant. Now they're having a emergent function that has an incidental effect compared to why they originated, which is that it allows you to tap into this new resource zone. And so, like, for example, the original reason that maybe some of the chemical metabolic substrates or the ability to absorb nutrients were not for the purpose of eating insects, right? But once you have the ability to eat insects, that actually has an incidental effect on your ability to expand in population and diversify. And that's relevant to the concept of innovation, because you're talking about the difference between just an invention and an innovation is that the innovation expands. And the concept of key innovation in evolutionary biology is usually something that repeatedly triggers diversification events. But I think you can separate.
Usually something that allows species to colonize a completely different niche, right? Something that wasn't available to them before. So in the sense of a pitcher or a carnivorous leaf, allowing these species to thrive in habitats where other species can't.
Yeah.
And as you say, it's very, very commonly not one single way or one single mechanism how these things come about, but there's multiple components coming together, and these components don't have to follow the same evolutionary path to get there, right? So the pitcher is a great example, because you already have the ability to take up nutrients, which you can see as a pre-adaptation. It's leaves in general have that ability to take up nutrients. But if you don't have a pitcher shape, you don't have that nutrient solution around the leaf surface most of the time. But as soon as you turn your flat leaf into a tube, into a funnel, you have the ability to hold liquid. That liquid can be nutrient enriched, so you get an advantage from that ability to take up the nutrients. You take up proportionally more nutrients to the leaf than before, because you now can hold this nutrient pool. Most plants secrete defensive substances in one way or another, or at least synthesize them, maybe don't secrete them, but are able to produce defensive substances that will attack chitin, so the insect exoskeleton. And it's been shown actually that the digestive enzymes of carnivorous plants have been co-opted from these defensive substances. So you already have selection for defense against herbivores that eat the plant. Now that you have a trap, these can be repurposed to help with the digestion. So
I was wondering about that when you first said it, because plants are basically fortresses.
Yes.
All these toxic chemical compounds, because they can't.
They're master biochemistry factories, right? So the vast majority of natural chemical compounds we know come from plants.
Has anybody tried to assemble a potential stepwise model of a series of events that would lead to the evolution, the convergent evolution of pitcher plants? I mean, because when you were just talking about it, it sounded like, okay, you have a key pre-adaptation, which is the ability of the leaves to absorb nutrients, and then you have to have a curling of the leaf, and so there could be some initial advantage to without the stickiness or the slippery surface or anything, just a curled leaf.
And yes, and there certainly is, because if you again think back to the bromeliads, to the tank bromeliads of South America, the vast majority of them are not carnivorous. They don't have slippery surfaces, but they have that reservoir of liquid that they hold in their leaves, and that accumulates debris that falls just in from the leaf litter from above, that just deposits from debris that floats in the atmosphere, right? And you end up with this nutrient-enriched pool of water in there. So pitchers most likely have evolved from water reservoirs to nutrient acquisition structures. And you find, when you look at carnivorous plants, you find the whole spectrum of things that are carnivorous, full-blown carnivorous, with all the traits that we associate with a carnivorous plant. And then you get some that don't produce their own enzymes, but they rely on organisms living inside the fluid pool. You get some that, need the help of other organisms to digest. You find some plants that aren't really classed as carnivorous, but they already have sticky leaves, and insects occasionally get stuck there and die and just get decomposed. And does the plant get an advantage in that case? Maybe it does. Very often the ability for all of that to be carnivorous is already kind of 80% there, but it's also the environment that the plant in that decides whether that is actually an advantage or not. So if a plant grows in a very nutrient-rich soil, being carnivorous doesn't give you an advantage at all, because the nutrients are there in the soil. So capturing insects, digesting insects is a fraction of what is available, right? So carnivory, and that's where we come back to where we started, right? Carnivory is only advantageous if mineral nutrients are limiting to growth. So if you don't have an abundance of sunlight and water and CO2, but we have an abundance of CO2 nowadays anyways, then being carnivorous is no advantage to the plant. Investing in trap structures would be a disadvantage if you don't need to be carnivorous, because a pitcher leaf is pretty lousy at doing photosynthesis. A leaf that's optimized for photosynthesis has a large area, and it's oriented at right angle to the light coming in. So a pitcher is very much the opposite of that. It's a vertical structure. It's often not particularly green anymore. They're either whitish or yellowish or reddish. Might have an advantage in attracting insects, maybe not. Maybe it's just saving by not investing chlorophyll in something that's not even horizontal. So it's always seeing these things in the right context. So another way of how evolutionary step changes can come about is just by taking an organism and throwing it into a complete different environment. And that's what we see in evolutionary history, right? The big diversifications always followed big step changes in the environment. There are glaciation periods, big changes in climate, big changes in environmental conditions, wet places turning dry, dry places turning wet, mountains lifting up, species becoming isolated. Yeah, so when we take an organism and put it into a new environment, then adaptation happens, right? Because then suddenly certain traits that are there and that weren't functional before become a big advantage.
Well, there's also just prior evolution creating novelties. Sometimes itself is a change in the environment that's so drastic that it triggers a cascade in other species. One example might be the evolution of C4 photosynthesis and the evolution of grasslands. And then that opens up the opportunity for all these new ungulate herbivores and then social predators like lions and things that hunt these huge herds of wildebeest and everything, that antelopes and horses and everything that eats the grass. And grass is different than big forest because the biomass is near the ground and it's digestible if you have the right adaptation in your teeth. And so you can get these. I do agree, that climate change and extinction events and all sorts of things have opened opportunities. But one thing that's also opened opportunities is just
evolution itself.
Yeah,
of course. The fascinating things about the natural world is that everything's interconnected, and the evolution of one group of organisms will impact on others. So it's changes in the environment are by no means limited to changes in the physical environment, but also changes in the living environment.
Yeah, so I'm trying to think about a theme from your study that I hadn't really thought about yet before. Was that and what you talked about in your discussion of pitcher plants more generally also just brings attention to, okay, you can have these different traits sort of randomly varying, and there's a role for kind of randomly getting the right combination. Like if you're just looking across species and they're varying across the species independently, then some get, the right combination of traits. And I was thinking about how that applies to what I was saying when I was studying kin recognition. I'm talking about a stepwise series of events that repeats itself. But it is true that for that series of events to happen, you have to have some certain conditions happen, being fulfilled. One of them being that you have to have a variable Q. The Q, whatever you're using to discriminate between kin and non-kin, has to be a variable trait. There's got to be some variation in it in the first place for natural selection to use that as a cue for discrimination. And so that's one of those traits that would be, you would need to randomly pop up initially for the whole thing to get started as a process. And then you would have to have the right conditions that favor the different traits along the way, like at the very first, the evolution of group living, some advantage to sociality. That's typically some ecological advantage, like ability to better hunt prey or avoid predators or build shelter, these kind of things that can come from living in groups. And then so on, you have to have the right combination of environmental circumstances and some of the initial traits that would let the thing happen.
I mean, variation is that you need variation is no new finding, right? I mean, that's at the basis of evolution. If traits aren't variable, they can't evolve. That's at the basis of any kind of evolution, whether it's selection driven or whether it's through random processes. You need variable traits for selection to act on, and you need variable traits to have something to combine in random combinations. So I think that's not a novel finding at all. It's maybe more the notion that you don't necessarily need selection driving towards a new novel trait combination, but that random combinations that occur just because of that variation being combined in random combinations can in itself be a powerful tool of generating novelty without the need to select. So selection doesn't have to come first. Selection can also come after a new combination has just occurred randomly. I think that's basically what our paper says. Right.
Yeah, I agree. And I think what's slightly confusing is that one of my traits, which is necessary for the complex trait to evolve in my case, a trait analogous to the horizontal springy lid of the pitcher plant is a trait, is variation in a trait itself, is the trait that has to be there. Which is, it sounds weird, right? Because you're saying in your study, the horizontal lid, once selection's acting on it, it becomes less variable because selection's acting on it. In my case of kin recognition, when selection's acting on this cue for association preference, it becomes more variable. And actually, the more variation of it is evidence of negative frequency dependent selection. So it's like the opposite, right? In my case, selection is evinced by a hyper level of variation that you would never get without selection acting on it to favor rare alleles.
In your case, then the trait that is under selection is not the actual trait, but the variability of that trait.
Yeah.
Right. Well, one of them. It's one step above. So you're in this case, the selection is acting like the positive selection is acting on actually making that trait more variable.
Exactly. Yeah.
Or keeping a certain variability of that trait, because if you lose that, then, but it's not the only thing that makes your kin recognition trait, right? If you just would have that variability without any of the other associated traits, it would be very meaningless.
Right. Yeah. That's just one of the cues of two different variable cue genes and two different discriminatory behaviors. So it's like altogether, it's a sort of complicated system with interacting discriminatory behaviors and cue genes. And one of the cue genes evolves to become variable adaptively in response to the other one, which is hypothesized under my model to have a low amount of variation that's maintained by extrinsic selection. But if you have that, then it can actually select for hundreds of alleles of the second locus. So you kind of have to randomly have the first one be variable, and each time selection uses a new cue at the outset, it needs to have some variation to just be used as a cue. But in this case of the second type of cue that evolves adaptively, after it's seized on by selection to be used as a cue, then it can evolve to become more variable. And then finally, when that whole system is in place, it evolves, according to my model, to avoid the immediate expression of conflict among non-relatives. But one of the effects of it that's incidental is that it can keep individuals from associating with non-kin or coming in contact with non-relatives. So if you think about an ant colony, and it has a nest defense system where it excludes any foreigners that don't share the same chemical profile. An effect of that is that it prevents transmission of parasites between the nests, interspecific parasites. That has nothing to do potentially with why it evolved, but that's an important effect sometimes. And you see that in invasive ants, where they've lost their natural kin recognition abilities, and they've evolved to be unicolonial in the introduced areas, but then they can transmit parasites, and they end up becoming susceptible to parasites that they're not typically susceptible to. And that's actually how people in the United States are doing biocontrol of invasive ants, is utilizing the natural parasites of the ants and bringing them here. And some of these studies show that, well, there's two forms of, like, the fire ant. One has the separate nests and one has connected nests. And the parasites only kill the ones with the connected nests that don't have the normal kin recognition between nests or colony mate recognition. And so there's potentially this incidental effect that might be important in the long run for ecological reasons. Okay, so I'd like to just mention one more theme, which is there's no these two examples of the spring trap mechanism are kind of one-offs. It's not like they triggered a diversification event. So there's no evidence yet that the spring trapping mechanism is an innovation per se. It's an invention. It's really neat. I think it's what David Attenborough might call a natural curiosity. I mean, but it didn't conquer the world, right? It's like found in these particular island habitats, and it hasn't gone anywhere yet necessarily. But the important, I mean, to me though, what we're really lacking an understanding of in studies of innovation is the study of invention, of really knowing how complex traits originate. And so the thing there is that you don't necessarily have to study the big innovations to get the crucial insights. And in fact, sometimes it's easier to study either the smaller innovations or things that wouldn't even be considered innovations, or just inventions or natural curiosities, to get insights into innovation. Because a lot of times with like the biggest innovations, they spread so widely, and they basically displace all the intermediate forms that connected them to ancestors, that it's difficult to make any inferences about where this thing came from, because there's no more intermediate forms connecting it to the past. So these examples where we can study more recently evolved traits, whether they're like in your case, an invention, or something that has potentially triggered some diversification event, but isn't so big that it totally wiped out everything that led to it. Those give us the best insights. And
I think it's just two different perspectives to look at the same process, which is the generation of novelty, right? So in our paper, we look at the generation of novelty from the standpoint of trying to understand how can that novelty have emerged. So it's an interest in the mechanism of generating novelty without looking at the future of that novelty. So whether that triggers a major diversification because it's a key step change in what species are able to do, or whether it doesn't, whether it just stays there as a novelty, or as you call it, an oddity, depends more on the implication of that new function for the species, right? So from an evolutionary point of view, it's one thing to generate a new function that's previously not been there from combining things that had nothing to do with each other. And then it's a second step to look at, okay, how does that impact then on the evolution of species, on speciation, on diversification, on adaptive radiations, and all of that. It's two different processes, two different things to look at.
Yeah, I think one study I would like to relate it to is okay, so the reason I think it's important to do these kind of studies like you've done is that it gives insight into the origin of novelties, like you said, which is sometimes difficult to study for the big innovations that happened a long time ago. And to give an example, one of the experiments that I really like that studied complex trait evolution was the Linsky long-term evolutionary experiment on E. coli bacteria. And this is a very well-known, talked about example. But I think it shows this what can potentially happen in the way we interpret evolution, and why we need these examples like yours. Because in that experiment, there was something like 5 or 6 steps leading to the evolution of citrate metabolism. Originally, the bacteria were not eating the citrate that was in the dishes. And I think the experimenters intentionally put it in there to see if they might evolve to eat it over time. At some point in the experiment, after 30-something thousand generations, I think it was like 33,000 generations, the bacteria all of a sudden started eating the citrate in one of the beakers, and they in one of the lines out of 12 lines, one of them started eating the bacteria, and the experimenter saw that the medium was cloudy, the bacteria had really grown in population. It's almost like the pitcher plant is now eating the insects, and so it's able to grow in population because it's getting into this new food source. And so because they froze all the samples in every generation, I don't know, every few hundred generations going back, all 30-something thousand generations, they were able to sequence the lineage and figure out what mutations were necessary and what happened. And there were about 5 or 6 mutations and selective events where the mutations spread and got fixed because of some immediate advantage in that environment. It had nothing to do with metabolizing citrate. And there was a key duplication at one point around 25,000 generations. And then past that point, there was a couple more steps. And only the final step or two kind of optimized to the citrate resource, to actually metabolizing citrate. But before that, it was like these things that were necessary for it evolved for other reasons, and then got co-opted incidentally into that pathway of metabolizing citrate. But here's a interesting thing about it, which is that one of the biggest mutation that was necessary, that was the most rare one, happened, I think, around 25,000 generations. And it happened in a very low frequency line that was so rare at that point that it was almost not detectable in that lineage, in that bottle lineage. And it was only once it had to gain the ability to metabolize citrate that it really expanded and overtook the population. Now, if you think about if this was like a species evolving in nature, maybe that thing would have spread, I mean, not just in that bottle, but like dispersed to the other bottles and totally displaced everything. So now if you're coming along later and you're looking at what's now present, there's nothing connecting it to the ancestor. And moreover, that's extant, right? Like you can't use comparisons between different species like you would in the pitcher plant example, or in a lot of these examples where you're looking for intermediate steps of existing species. No, you just have the one thing. It'd be something like the genetic code, right? There's like almost nothing connecting it to whatever the former code was. There's just like every organism on Earth has the same genetic code except for a couple of subsequent modifications. So you don't have like any evidence of the transitional steps. And then the other thing is, at that point when that important mutation happened and the steps after that, it was so low frequency that if you're talking about studying the fossil record, organisms that are rare don't leave many fossils. So you're unlikely to find that in the fossil record. So then what happens, what would happen in that case is all of a sudden you would just see the final complex organism able to metabolize citrate suddenly appear in the fossil record, because before that point it was so low frequency. It was evolving, right? And it was there, but it was so rare that if it was something that you're looking for in fossils, you would never find it. And so then it looks like it is saltation, that it all these traits came together really quick all at once in this one thing, even though it was evolving gradually over time. So for that reason, I think when we've actually looked at the fossil record, we've often seen the sudden origin of higher taxa and an apparent like sudden burst of evolution. Gould called it punctuated equilibrium. What causes this discontinuous change in the fossil record? And that gives a potential, people have often said, well, it's saltation, right? There's something happening really quickly in genetic revolutions, when in reality it could be more or less gradual evolution. And then the reason it looks like saltation is that you're not seeing the fossils of the intermediate forms, but they did exist. And And so it just makes innovation hard to study in those examples, which is why the more examples of invention of really looking at not just accepting the standard reasons for why complex traits evolved, but doing like you did and really being curious and saying, well, look at that. That's interesting. How did this composite trait come about? And you didn't just leave it at that. You went and said, well, how can I study this? And you talked to people and you figured out a way to approach the study that was very different than what people have been doing in the past. And there just really hasn't been many examples of that. I mean, the folder I have about complex composite trait evolution, where there's actual evidence of what happened, it's not many papers. It's a very small literature, and it's funny because people talk about Darwinism, and well, Darwin's great feat was to explain design and nature and complexity of design. But ever since Darwin, people have been focusing on simple traits. And it's like, okay, well, how much have we really gained in understanding that since that time? I mean, Darwin threw out a couple of examples, the eye, and the other big one was the evolution of the honeybee honeycomb. And he spent a lot of time thinking about that problem, because at the time that was one of the examples where you've got this perfect trait, and well, it must have been created for this final purpose by a creator. And so he spent a lot of time trying to figure out how that could have evolved gradually. But other than those, a few examples, it's like there's really not. I mean, I've looked into this. I mean, it seems like to me that you study these pitcher plants, and you just saw this as an interesting question. I don't know. I mean, are you aware of like how rare and this type of study is in evolutionary biology?
I mean, we obviously started, especially me as being not an evolutionary biologist, started like digging into the literature and trying to find out what is known. And yeah, I came across somewhat similar problems like you, that I found that there is an awful lot of theoretical papers that try to explain complex trait evolution, and there's surprisingly few studies looking into analyzing actual data. And as you say, it's difficult to go back in time. If you have something very complex, we can't do these studies unless we do them with bacteria. And those studies are very, very valuable, and they just come from a completely different angle. I would say they're just as valid and just as valuable as what we've done. It's just a different way of looking at things. And I believe there's a lot to be gained from looking at problems from very different angles. So maybe there was an advantage coming from a different background and looking from analyzing the phenotype that we see right now point of view, while a lot of studies try to analyze evolution at a genetic level, which is just as valid. It gives a different insight into the same topic from a different point of view. So I think the more points of view we combine, the more we learn. And the more data and systems we bring together from different disciplines, the more we learn from it.
Yeah, well, I was trying to figure out if your group of authors, beforehand, I didn't know whether it kind of gradually came together or whether it was spontaneous coincidence.
It was more of a gradual building of a team, I think. But still, there were a lot of random effects involved in who you meet when.
Yeah.
So that applies certainly to everything. I think there is a lot of discussion in evolutionary biology going on, like whether Darwin's view of seeing the world explains everything, or whether we need to add to that. We've deliberately taken a step back and tried to not get involved in that big and heated discussion amongst evolutionary theoretical researchers, because we feel I feel certainly not qualified to contribute to that discussion. I found a really interesting paper in Nature from 2015 where there were two schools of thought face off against each other. There's the purists who just say everything since Darwin, since the modern synthesis, is enough to explain what's going on in evolution. And then there's a big field of evo-devo people who say we need to look beyond that. We need to look at developmental processes, at plasticity, at epigenetic effects, and how that plays into evolution. All the things we learned since Darwin. And I've read it with great interest. I don't feel confident or competent to comment on who is right. I think they probably both have very valued arguments, and I really liked that the article finished with a final paragraph, which was the only thing where both sides agreed, which was the point that there aren't enough data, that there's a heated discussion, but there aren't enough empirical studies looking at evolution of complex traits. And we thought if we can do a tiny little bit to contribute to that and to provide some more data to fuel that debate and to give people who know way more about evolution than I do something to talk about and something to make up arguments from them, we've done more than what we wanted to achieve.
Well, it might not be the case that the big controversies are indicative of the what the next new thing is. It could be something very low frequency happening in obscure places that is going to take over. And I think a lot of the big innovations in science, it's not what people are talking about at a time. It's some lone person or small team of people that totally think outside the box and then come up with something completely new. And it totally steps outside the bounds of the normal controversies and everything. I personally don't think that those controversies you're referring to are really like touching on the key issues. And I feel like it's people that really don't disagree with each other in fundamental ways. They're all adhering to the same shared assumptions. And so I think their controversies aren't really like relevant to what the big advances will be in the future. But I do think that your paper is relevant, and we need more data and studies of complex traits like yours. Is there anything else that we've missed?
I don't think so. We've talked for quite a long while.
Yeah. I've actually got something at 9:45. So, Ulrike, thanks for being on the Natural Reward podcast. I really appreciate it. This was a fascinating discussion.
Thank you very much for having me, Owen. It's been a pleasure. Nice to meet you. Bye.